Recording here from the Rollins School of Public Health at Emory University, this is the Virulent Vortex, a podcast in which we discuss infectious diseases from molecules, pathogens to populations, pandemics, and everything in between. My name is Jaap de Rode. I'm a biology professor here at Emory, and with me today is
Deanna Kulpa. I'm an associate professor in the Department of Pathology in the Emory School of Medicine, as well as being an investigator in the Emory National Primate Research Center in the Division of Microbiology and Immunology.
Well, thank you so much for being on the show today and talking about your research. My understanding is that a lot of your work focuses on HIV and understanding how HIV infections can last as long as they do. So I wondered if you could start by explaining what HIV is and then also how you became interested in studying it and the specific questions that you look at.
So yeah, absolutely. My lab focuses on the study of HIV, which is a virus that's been around for a long time, although we first started realizing that it was infecting people and causing a problem in the 80s. And now we understand that it's a virus that impacts the immune system. Specifically, the virus will enter into immune cells, CD4 T cells, and then it lives in those CD4 T cells. So part of the problem that the reason why we can't get rid of it is it's not only a problem in terms of the immune system trying to identify the virus itself, but it's actually inside of the immune system cells that we need to actually fight the virus. So what happens is HIV is killing those very cells that we need to go on and combat the infection. So that's one part of the problem.
So that's a really big paradox, right? We have immune cells helping us to protect against viruses. But here you have a virus that goes inside those immune cells, hides in there, but also destroys them.
Exactly. The other aspect of the problem is that, at first we thought, okay, how do we find drugs or compounds that would be able to stop viral replication? Because if the virus is going into the immune cells and killing immune cells, can we stop that process? And so we developed antiretroviral therapy, which is just different compounds or classes of compounds, but actually interrupt viral replication itself. So now we've halted that process, but we now understand that the virus, once it gets into a cell, so once it gets into those CD4 T cells, it's a permanent resident. So essentially, every virus has a genome, genetic material that carries a blueprint for making more viruses. And this particular blueprint is one that actually inserts itself into our own genomes inside of our own cells. Mm-hmm. And once it's in there, it's a permanent resident. So that cell that's carrying that viral DNA, it sticks with it for its entire life. And so part of the problem of that is that once it's in there, these are actually, our immune cells are meant to last us for an entire life, right? You don't stop having immunity when you're 50 or 60, right? We have immunity for our entire lives. And so these are the very cells that the virus is getting into. And so these are cells that are meant to last your entire life. Yeah. And so getting rid of those cells is very problematic. And it's really the focus of what my lab studies, which is HIV persistence, which is just the fancy way of saying, how do we understand how that virus lasts forever.
Yeah. And so to clarify, so these CD4 T cells, they're a type of immune cell that is normally involved in helping control these infections with viruses. And so when you say they last a lifetime, is that individual cells or do these cells replicate? And if they replicate, do they also replicate the viral DNA that's inside them with them?
That's a very good point. So it's exactly what happens, right? So it's not an individual cell that just sticks around forever. What happens is our immune cells basically go through cell division over time, just to kind of maintain themselves. And as you just said, a cell that has the virus in it is then going to replicate that virus as it replicates itself. And that's just a way of the immune system maintaining its memory, right? So how memory in the immune system works is a virus that you're exposed to when you're 10, we develop an immunological memory to it. So we've seen that virus once, and since it's in our environment, we'll probably see it again. And so what we want to do is be able to maintain a memory to that virus so that as you encounter it, when you're 20, 30, 40, you're able to respond to that pathogen rapidly. And that's what our immunological memory does. And those are the very cells that HIV hides in, is those cells that are meant to kind of respond to those environmental challenges over and over again in our life.
And so it's hiding in these cells, and then does it ever come back out?
It can. So one of the things that we know with viruses that establish a latent infection, which is what that is, just a virus that's silent when it goes into and integrates into the host cell genome. What we would expect is if it's going to make more virus, it's got to express viral proteins, right? It's got to put all the components together in order to make that virus, what we think of that particle when we see images of that goes out of the cell and finds a new target to go into. In order for that to happen, the virus has to express those proteins. When it's not expressing those proteins, it's silent or latent. And so what we're interested in is what makes a virus silent, what makes the virus express proteins. And one of the things that the immune system needs in order to identify a cell that has a virus in it is it has to signal that there's a virus there. And the way our cells do that is by taking bits of those viral proteins and putting them on the cell surface. And that's kind of a flag that says, okay, I'm infected, come do something with me. And so the other immune cells can come and recognize that there's a problem with that cell in order to get rid of it. But if the virus is silent and not expressing any of those proteins, there's no flag. And it just that cell just kind of goes around undetected. What happens is that our immune cells, they're specific to different pathogens that we encounter, right? So think of it as an army, that there's all these different divisions and all these different types of cells that are responsible for making up that army. So when you have a particular situation where you need, your battalion or whatever, then you're going to call out that part of the army. So if the virus is hiding in those cells that are in that part of the immune system that we need to when we encounter flu, when you're exposed to just something in your environment, you have a cold, then when that part of the army gets called up, then those cells become activated, and that's when we start to see the virus expressing again. And so there's an opportunity to be able to recognize those cells because they're going to be expressing viral proteins. The problem, of course, HIV is a very complicated problem, is that because of the impact that HIV has had on the immune system, it doesn't work very well. Basically you've already hindered it. And so, to be able to then say, okay, how well can we see those cells once they are waving that flag around? It actually doesn't work as well as it would have if the person had never had the virus in the first place. So it's a very complex problem, and it's one of the reasons why it's taken us so long to kind of make the sort of progress we have.
Yeah, and so in your own research, so what have you found about how these viruses can persist this long? Because that is really one of the key questions, as you mentioned, that you're really interested in.
Mm-hmm. So one of the things that we have found is looking at what are the mechanisms that turn on virus in a cell. And so one of the things that we've been studying is kind of all aspects of a cell in particular. What does it look like? How does it work? And one of the things that we've become very interested recently is cellular metabolism. So metabolism is a word we've heard, a lot. We think of people who have a fast metabolism or a slow metabolism. The people who have a fast metabolism, they can eat a lot. They've got a lot of energy. We think of people with a slower metabolism. Those are people, maybe they'd like to have a faster metabolism. So what we understand now is that cells have their own metabolism too. And so when we think about cell metabolism, the cells that have a high metabolism, let's say, are very active and function very well. And what we now understand is that aging is one of those things that can slow not only our whole metabolism, but it also can slow cellular metabolism. So over time, we see the components of the immune system that we're interested in, which is CD4 T cells, their metabolism slows. Well, one of the things that HIV does is it accelerates that slowing of the metabolism. So essentially, it makes their immune system look older than it would chronologically, according to the age of that person. And so we're interested in cellular metabolism because when we think about, okay, we want to target that cell in order to, activate the virus so we can see the viral proteins, the slower the cellular metabolism, the less it's going to respond to those types of signals, the sort of the dampening of the response. And so we want to be able to understand why it's doing that and then what we can do about it. Because not only can this tell us about HIV and cell metabolism. But it also gives us a clue about aging and cellular metabolism in terms of how we can apply those same ideas across not just HIV, but the thought of aging in itself and how cellular metabolism slows and what could we potentially do in terms of helping to maintain a higher metabolism, not just in our person, but in our cells themselves. So what I like about the research is that even though we're studying something in this particular state, it has applications across, many different disciplines and can give us an insight into potential ways we can help ourselves over time, stay with a higher metabolism, be able to respond to things quickly. And so that kind of is the insight we're looking for.
So in terms of this aging, and some of us are doing that right now. And so, and I can speak for myself that aging means things become harder, not as good at certain things. And so that's the same for immune cells. But then the other question I have is, could you also use that? Can you recognize cells because they are older? And is that a way to identify where the viruses are as well?
That's actually an excellent point. And that was the focus of research for quite a while. And we actually borrow a lot from cancer research, for example. So there's a lot of overlap between cancer biology and HIV biology. And so things that work in terms of the cancer field, because what we see in cancer is if there's a cancer present, why isn't the immune system taking care of it? What is it about the cancer state that is impacting the immune cells' ability to recognize and deal with that? It's the same kind of thought with HIV. Like, we have a virus there. Why isn't the immune system able to recognize that? So there's a lot of crosstalk between the two disciplines. And so what tends to work in terms of compounds or things that are tried in terms of agents for treating cancer, we actually try those in HIV as well. And so that's something that we see when it comes to aging of the immune system. There's compounds in cancer that actually work well in terms of HIV in order to be able to do that. And that is sort of like thinking of resetting the clock in terms of kind of dealing with those pathways. What we found in our research is if you address it that way, if you're looking at some of those compounds that are trying to reset the clock, you still haven't gone deep enough, essentially. And that starts back to what I was saying about cellular metabolism. Like you've sort of like got the top layer, but you actually need to go down into the heart of like what makes the cell tick. And we're finding that's really what is one of the drivers for HIV persistence in particular.
Yeah. And my understanding is you also do work with human clinical trials. Is that right? Yeah. So can you tell us a little bit about that?
So most of the work that we do, so I'm part of the Center for AIDS Research here at Emory, and we have work with some clinicians who are part of the Center for AIDS Research as well, who are able to recruit participants for us to be able to get some cells from the individuals and be able to do experiments in the lab. What we do that for is because what we can do basically in a dish in modeling, the sort of cell interactions that we're looking for can only get us so far. So being able to then go into individuals and be able to ask them, is this what's happening in a person? And one of the things that is very interesting to us in terms of persistence is being able to look at people who've been living with the virus for different amounts of time. So I was talking about aging and how the virus accelerates aging. Can we look at individuals at different periods of time who, been exposed to the virus or been on antiretroviral therapy and see how that's impacting the immune system over time, and basically allow us to see exactly where therapeutically, we need to intervene. Because one of the things when we talk about metabolism, maybe someone who has been exposed to the virus for a shorter period of time, that is going to maybe look differently therapeutically because their immune system has not had that long-term exposure, has not had that sort of accelerated aging impact. But now we've had people who've been living with the virus for 30 years. Their immune system is going to look very different. And that's a significant population now. Thankfully, we've had people who've been able to live with the virus because of antiretroviral therapy. But their immune system is quite different. And so we are looking at those people as a way of understanding how do we need to address these different populations when we're thinking about a therapeutic intervention? Because our research is suggesting that what's going to work for one population may not necessarily work for another. And what I find is when we're thinking about studies that have been done so far, they tend to kind of group everybody together, right? They're basically saying everybody who has HIV, let's go into the clinical trial, and not really looking at people as far as maybe a personalized medicine sort of approach is kind of like how you might think of it. And so what I'm very interested in is saying, okay, can we look at that data and then look at people basically kind of trying to bin out, like maybe we have some people in there where that therapy worked really well, but because, everybody was looked at as a group, you didn't see that.
Because I imagine when you do human trials, often you may look at the age of the person, but you're basically saying we need to look at the age of the infection.
Yes.
That's really intriguing. And really thinking about HIV as a major pandemic. Sometimes we forget that we have other pandemics taking hold in our daily lives. But of course, HIV has been a pandemic for decades. That on the one hand allows you to study these things, but also really stresses that we still need to figure this out and try to find the biological ways to help people.
I mean, right now, there are 39 million people living with HIV, globally, which is a pretty big number. And one of the things that I think people, maybe particularly in the United States, don't realize is over half of those are women and girls. And one of the things that we're interested in as well is when we talk about this metabolism of aging, we know that the immune system of women is different in terms of, especially in HIV response, but it's different than men. And a lot of what we've studied in the United States has been men, particularly in the context of HIV. And we know that bias exists for a lot of different, medical studies, but in HIV in particular, it's been men. And so we've been working with our community partner here in Atlanta, Sister Love, who is a women's reproductive rights organization, and they do a lot of advocacy for women with HIV. And so partnering with them has really kind of made sure that we're aware that women need to be one of the things that we focus on, and understanding the immune system in women living with HIV as well, because it's half of people living with HIV right now. We can't forget them.
Yeah, and that's a lot of people. Yeah. Well, this was so interesting. Thank you so much for sharing your research with us.
Yeah, absolutely. It was a lot of fun.